Okay, I'd like to thank the course directors for the opportunity to come and talk at this meeting and also to make my third visit to Egypt. So in the next 20 minutes or so, I want to try to introduce some of the concepts of mechanobiology and how they affect and how they're involved in the pathogenesis of the atherosclerosis process. Indeed, there have been a number of theories and questions addressed that have come up with possible explanations of how the atherosclerotic process evolves, relating to the accumulation of lipid within the vessel wall, material from blood deposited from the passing flow of blood, and also the Russell-Ross response to injury theory, in that the damage to the endothelium causing effects on underlying smooth muscle cells and release of polypeptide growth factors. However, when you look at the distribution, or the classical distribution of atherosclerotic lesions, researchers back probably almost as long ago as 50 years ago saw that the lesion formation, particularly at branching points and in the curvature of the aorta, suggested that indeed fluid dynamics may well have a influence to play on this process. And indeed, the early thoughts, again going back to the early 1960s, related to theories that flow separation may occur within the arterial system, and that this may indeed occur at branching points, and that this can cause mechanical damage to the vessel wall, causing platelet deposition and fibrin deposition. But really, the early studies made no concerted efforts to attempt that their suggested sites of wall damage with the actual sites of atherosclerosis. And indeed, they thought initially that high shear strains were likely to be the cause of the atherogenic process. However, this work was in some way undermined by an incomplete understanding of fluid dynamics. And it wasn't until a group at Imperial College, led by Colin Cairo, in fact, suggested that the curving and the branching of arteries can promote low shear stress at certain points within the vessel wall. And they predicted, in fact, that the regions where they would expect this to occur were, in fact, where the atherosclerotic lesions would occur. And indeed, they were able to correlate these two phenomena, suggesting that low shear stress is a primary event in the atherogenic process. So just a little bit about fluid dynamics, and I'm no expert personally, but shear stress is caused essentially by the frictional force of the blood passing over the endothelial cells that line the surfaces of all blood vessels. And really the shear rate is the product of the blood viscosity and the spatial gradient of blood velocity against the vessel wall. And therefore this can in fact be calculated quite straightforwardly. Different types of flow may occur in the vascular tree. Either the so-called undisturbed laminar flow, where we have a streamlined flow pattern with concentric layers of blood moving in parallel lines. And this will cause a high shear along the endothelial surface of vessels. In cases of branching or obstructions, this flow may become disturbed or disturbed laminar flow and can be characterized by, in fact, reverse flow in certain areas and areas of stagnation. A third type of flow, which doesn't really occur and has no significant effect on the atherogenic process, is turbulent flow, where you have almost random motion of flow after some sort of blockage. But essentially what Cairo and colleagues are able to show is if you take a branching point where the flow divides, you'll see on the inner surface of this vessel wall a maintenance of high shear stress. However, on the opposite surface of where the branching occurs, you get this recirculation of the flow and eddies forming and points of stagnation, such that the shear stress on the inner surface of the vessel wall will be high, with a peak with the pulse of the blood, which will then reduce until the next pulse wave comes forward. However, in these areas on the inner surface, on the outer surface, sorry, we get much less shear stress, and we get a forward and a reverse flow. And it's these areas that we're believed to then, or have been shown to be the areas in which atherosclerosis preferentially occurs. Indeed, not all flow is the same, and not all flow will produce, or levels of shear stress will produce similar responses. And this is really a caution to in vitro experimentation along these lines, in that these types of single impulse or a ramped pulse may have the same numerical value of shear stress, but will in fact be capable of producing completely different types of responses. And indeed, this is an example of such an experiment where we have this ramped flow and a pulse flow. And in this experiment, reported in an excellent review in the Philosophical Transactions of the Royal Society by White and Frangos, this molecule, which is a fluorescent molecule, was incorporated into the membrane of endothelial cells, and actually will signal as changes occur in the viscosity of the membrane. And you can see that the changes in viscosity will occur in parallel to the changes in shear stress, whether as it increases and decreases, as with the ramped flow. So the amount of changes in the vessel or in the endothelial cell membranes can vary according to the type of impulse that the vessel wall sees. And this is actually quite intriguing when you consider some of the more recent non-pulsatile LVAD circulatory support devices. And it'll be interesting to see how these people's endothelial cells actually respond to having a non-pulsatile circulation. In generating flow, there's also a change in the tension experienced by the endothelial cells. So as flow goes in one direction, the cells will generate tension in the opposite direction as they resist the flow. While they do attempt to line up in parallel to the flow, these tensional changes are also thought to be important, and that it's been shown that they're sensed by cell junction molecules such as PCAM or CD31, and these can be associated with G protein coupled receptors. And again reported and described in that review is very nice work in showing how these two molecules may act together, and in fact that you can then induce signaling in endothelial cells by these tensional changes. Indeed, there are a number of potential different signaling mechanisms rather than just tension changes. We can also see again the changes in membrane fluidity caused by shear stress, as described in the earlier study with the fluorescent probe. And it's also been shown that just this change in membrane fluidity allows activation of G protein coupled receptors. And therefore, this allows activation of these receptor signaling proteins without any ligand binding as such. So these again are thought to be potentially important mechanisms that can regulate and control the response of endothelial cells to shear stress. So if shear stress is going to change, the magnitude of these signaling events will in fact change. So going on to those changes in membrane fluidity and therefore potentially changes in stiffness and the mechanical stress that endothelial cells see, it was interesting to see a recent paper published by a group at Imperial whereby they looked at regions of the inner curvature of the aortic arch and the outer curvature, and imaging these cells with a scan ion conductance microscope. They were able to show a difference in the morphology of these cells. So they're being affected differently by the different types of flow that they were seeing. And indeed, when they then quantified this cell shape change, the elongation in the cells in one area was significantly different to that in the other area. This is on the inner curvature, whereas the greater degree of elongation occurs where you see the outer curvature where there's the high shear stress. And this was also associated, or these cells were also associated with difference in membrane compliance or stiffness. So again, the stiffness of the cells changes due to the type of flow that they're able to see. And again, this may all work together to influence the different types of signaling mechanisms that can be induced by either low or high shear stress. So essentially, the field has progressed to define these areas as atheroprone in low shear oscillatory flow areas and atheroprotected areas where you have the high laminar shear stress. So it's interesting then to see perhaps how shear stress may therefore actually affect the function of endothelial cells and how this may be coupled therefore to pro-atherogenic mechanisms. And indeed this cartoon shows, it's rather complicated, but shows that shear stress or low shear stress on an endothelial cell can induce a whole range of signaling mechanisms, with just drawing attention to the fact that shear stress is an important signaling stimulus for the release of endothelial-derived nitric oxide, which may induce changes in how cells attach to the endothelium or changes in how it signals to the underlying smooth muscle cells. It also changes the amount of oxidized LDL that can be taken or released and can be taken up by the smooth muscle cells. As we heard nicely in the previous talk, matrix degradation, MMPs can change, can be increased. Inflammatory mediators such as MCP1, the cell adhesion molecules, VCAM1, ICAM, E-selectin, as well as the release of cytokines, can all change in circumstances of low shear stress. Procalcification mediators such as the bone morphogenic protein 4 can be released, and also polypeptide growth factors in line with the Russell-Ross theory in response to injury, such as PDGFs, FGFs, and endothelium-1, which may go on to stimulate smooth muscle cell growth, differentiation, and angiogenesis. So the question is, how do endothelial cells actually manage to sense the change in flow? We've looked at some aspects of changes in stiffness, changes in membrane fluidity, but there's also a whole range of potential sensing molecules and proteins that are expressed on the surface of the endothelial cells. And really I want to concentrate mainly on the glycocalyx. So the glycocalyx is essentially a, if you like, almost a forest of proteoglycan-like molecules that stick out on the surface of endothelial cells into the blood. And these, this glycocalyx is able to sense the flow of the blood and is linked to the signaling of nitric oxide synthase. So we can see this on various EM pictures. We can see as like almost sort of hairy projections on the surface of endothelium. So in a quite elegant series of experiments published in 2007, a group in New York attempted to remove the glycocalyx and these proteins selectively, either by removing the heparin sulfates, the salicylic acids, or the hyaluronans. And here we see the response to shear stress in the first bar. And then with the heparin removed, we see a reduction, the static control, and the static control is not changed with the removal of heparin. And in the same way, we see changes again in the amount of flow-dependent NO release. This is measuring accumulation of nitrate in the media. We see a reduction in the flow-mediated responses. Interesting, there are many other ways of stimulating nitric oxide, principally by adding a number of different vasoactive agents, one of which, as they chose for their experiments, is histamine. And when you repeat the same experiments, but you stimulate nitric oxide with histamine, these responses are unaffected. So this is entirely a flow-dependent mechanism for sensing and causing the release of nitric oxide. Also, changes in the shear or areas of low shear stress are also associated with changes in the transport of molecules through endothelial cells, such that these low shear stress areas are associated with a loss of tight junctions between endothelial cells that then allow transport of micromolecules through these gaps into the underlying vessel wall. And then going on to inflammation, again, nitric oxide can play a role here, but also the expression of the cell adhesion molecules, such as selectins, the release of chemokines and vascular adhesion molecules. When they're expressed upon the endothelium, this is how then leukocytes are able to initially become tactile for the endothelium. They start to roll along it until they eventually become firmly attached and then undergo transmigration, causing the inflammatory response to transgress into the underlying smooth muscle cell. And indeed, in terms of inflammatory pathways, and again, we heard in the previous talk about the NF-kappa B pathway, but also cytokines can stimulate other inflammatory signaling molecules, such as the MAP kinase pathway. And it's been shown quite nicely by a group based at the Hammersmith Hospital that shear stress, whether it be high shear stress, inhibiting directly some of these inflammatory signaling pathways, NF-kappa B or MAP kinase, or in fact stimulating inhibitory molecules onto this pathway, can end up regulating or preventing pro-inflammatory transcripts for the expression of other inflammatory molecules, such as the vascular adhesion molecules. So conversely, when this high shear stress is reversed to low or stagnant areas of flow, then these protective mechanisms are removed, and this allows inflammation to be promoted and to continue unabated. I just thought I'd diverge slightly and just give you a few examples of how shear stress also affects other calcification or atherosclerotic-like mechanisms, in terms of there are certain lessons to be learnt from the aortic valve as to how flow can be influenced. So the aortic valve is quite a nice model, because on one side, as the valve opens, you get high shear as the blood is ejected from the left ventricle, and then as the valve closes, you get this disturbed pattern of low shear stress. And unsurprisingly, perhaps, aortic calcification, aortic stenosis, the calcification lesions tend to always occur on the upper surface of the area, of the upper surface of the valve, where we see this low level of shear stress. And indeed, when you look at the genes expressed by these endothelial cells, this is quite a busy slide, but essentially you have the lower expression of anti-calcification genes on the aortic side, and you get a higher expression of pro-calcification genes on the aortic side. So there's a predisposition of these cells to undergo aortic calcification processes. And indeed, you can also look at changes in endothelial function. This is measuring cyclic GMP. This work carried out by Ismail Hamamsi, looking at differences by stimulating the endothelium on the aortic side or the ventricular side. and the aortic side appears to release less protective nitric oxide. Also, you can look at the strain experienced by endothelial cells and show that if the strain increases, this also can produce a pro-inflammatory state by inducing the expression of cell adhesion molecules. Again, Ishmael, when he was at Harefield, also went on to look at how the endothelium may in fact be linked to stress distribution, since these calcification nodules seem to occur where there are high levels of stress within the valve. And in fact, he was able to show that if you added sodium nitroprusside, which is a nitric oxide donor, or 5-HT, which releases nitric oxide from valve endothelium, that this was able to reduce the stiffness of the aortic valve tissue. So therefore, the endothelium appears to be able to locally regulate the amount of stress in the valve. So this is, again, a potentially interesting mechanism that perhaps doesn't translate directly to the vessel wall, but is more analogous to the dilator effects of nitric oxide in the coronary artery wall. So I've just chosen a couple of experimental examples of how some of these phenomena can be studied. And in this ligation model, whereby in the two carotid arteries, the left carotid is ligated and the right carotid artery is left untouched, this colour map shows the level of shear stress pre-ligation and post-ligation. So here you can now see in the left carotid artery we have very low shear stress indeed compared to the right. And this again, in the same way, in the same manner as the valve, is associated with gene changes in gene expression, which have been shown to be pro-athogenic in their nature. Lastly, this study that looked at the sort of natural history of the atherosclerotic process in a porcine model of atherosclerosis. And here if we look at sections 1 and 4, we see increase in the presence of atheroma, staining for oil red O and CD45 is increased in these regions. And these were associated with areas and points of very low shear stress. So there seems to be a good correlation in experimental models as well as what is observed in humans. So if we look at the progression of plaque formation and perhaps thrombus formation, really in the time scale of this process, the role of mechanotransduction and the influence of flow on the endothelium is really attempting, or the high shear stress is attempting to control these mechanisms in the very early stages of this process. And there may be events, risk factors, for example, that then tip the balance in areas of low shear stress to allow for these inflammatory processes to come into play and to predominate, and for the process to progress. So just to summarize how shear stress may affect different types of lesion formation. So low shear stress may cause an early atheroma. Some of these progress to remodel in a way that physiological endothelial shear stress is maintained and there is limited inflammation, and this person would therefore remain asymptomatic. Sometimes the fibroproliferation was into the lumen, where you then actually would then have a region of high shear stress across this area due to a constrictive remodeling of a stenotic plaque, and this patient would then essentially remain stable. But since all of these are interchangeable, if you then get an excessive expansive remodeling and this area expands, you'll then reduce the amount of shear stress, and this plaque may then fall into the vulnerable category and experience intense inflammatory reactions and may eventually rupture and lead to an acute coronary syndrome. So I'd just like to thank you for your attention. This is the Heart Science Centre on a nice sunny day, not like when I left it, and I'm happy to take any questions. Thank you.
Thank you very much, Adrian, for such a clear review of the subject. I'm sure we will hear a lot more about mechanobiology in the future.
Thank you, Dr. Chester. Actually, you modernized what we knew when we were in medical school, that the shear effect is important in the pathogenesis of atherosclerosis. But why shear effects occurred, for example, in coronary vessels, and the cerebral vessels and lower limbs are perfect, and the vice versa? Why it chooses certain arteries and not, although he's the same patient with the same condition and everything?
That's a very good question and difficult to answer. I think some of the localized effects that may cause a precipitation of atherosclerosis, or not as the case may be linked to actually changes where endothelial cells start to proliferate and change locally, and thereby you may get local changes that then disrupt the pattern of flow and lead to the stimulation of some of these signaling molecules or reductions of nitric oxide. How and why they occur in certain areas, I think, is a difficult thing to pin down.
May I step in here, Adrian, with your permission? I think there is evidence that the endothelium is not the endothelium, is not the endothelium. There are site-specific differences in the endothelium, which is specific to organs, specific to regions, and vary in different subjects, and some of it to do with lineage as well, where they came from, like the descending aorta is more vulnerable to atherosclerosis, while the ascending aorta is more vulnerable to dilatation. And if you take the descending aorta and put it in the ascending aorta, it still has the same characteristic. It will still go atherosclerotic. So mechanobiology is one thing, but lineage and endothelial specific characterization to the to the organs. We have questions from the floor. Professor Newby.
I've got absolutely no problem with low flow dictating where lesions begin. Massive evidence for that. I've got a bit more of a problem in understanding whether the low flow is that much involved in the transition from stable to unstable plaques, because say, for example, in the carotid circulation, we know that the most unstable plaques are more than 70% stenosed, and they should have, as a consequence of that, massively increased shear stress over the top of the lesions. And in addition to that, most of the lesions occur on the upstream side, which is the side which experiences the highest shear, not on the downstream sides where you might get disturbed flow. So no problem about initiation, but there must be something else that intervenes later on in the more advanced lesions.
I agree. I didn't mean to intimate that it was all down to flow, but clearly there are other mechanisms that are probably important in modulating, as you say, the transition between different types of lesion. I agree with you.
Yes.
Well, it was very interesting, and I think all of us, or many of us, know that shear stress, there is strong evidence of its effect on atherosclerosis, but my question is about the implication of the presence or absence of hypertension. Because many epidemiological data show that hypertension is maybe the first or second important risk factor in atherosclerosis. There is correlation between hypertension and the effect of stress on the artery?
That's an excellent question, in as much as my talk only really set out to address the mechanobiology of the blood on the endothelium, but of course the distending effects of hypertension felt by the vessel wall would also change the mechanical environment that a vessel wall is experiencing. So in one of the slides I showed on the valves, when they have increased stretch, they become more pro-inflammatory. Then that could also be the case for how the endothelial cells and or the smooth muscle cells may sense an increased distension caused by hypertension. So it's an increase just as an important mechanism, but my talk didn't focus on that specifically, but you're quite right to bring it up.